Hi everyone, I am Lynda, a senior at Georgia State University majoring in chemistry with a concentration in biochemistry, and I am presenting a study of near-infrared DNA damage and photo-cytotoxicity by a brominated 4-quinolinium dicarbocyanine dye named ESS24. My research focuses on photodynamic therapy, or PDT, which is a form of therapy in which a photosensitizing agent generates reactive oxygen species in cancerous tissues. This process will damage DNA and other macromolecules, which will lead to cell death. PDT is effective against ailments for which surgery is not an option. Many PDT agents usually absorb red light. This is because this wavelength window allows for light to reach deeply into cancerous tissues. Photosensitizing agent is an agent that has the capacity to oxidatively cleave DNA. They are activated via excitation with low energy irradiation, which causes especially focused oxidative damage to DNA and other macromolecules in cancerous cells. Here I work on a specific dicarbocyanine dye named ESS24. ESS24 has demonstrated through its characteristic that it could be a potential candidate for PDT. It is also taken up by ES2 ovarian cancer cells and causes a substantial level of near-infrared photocytotoxicity. Carbocyanine dyes are fluorophore that are used in vivo imaging. They are compounds that contain 2 nitrogen atoms linked by an odd number of methane units. In my case, there are 5. Also, the nitrogens are part of the heterocyclic ring. As you can see in figure 1, ESS24 is composed of 2 aromatic rings which contain nitrogen bonded to methyl groups. The 5-carbon chain that links the 2 aromatic rings containing the nitrogen has a bromine at the center. So it is mesobrominated as well as conjugated. Figure 1b shows the mechanism of photodynamic therapy, which requires both a photosensitizing agent and light in order to create a reactive oxygen species. Examples of reactive oxygen species include hydroxyl radical, superoxide, nitrogen peroxide, singlet oxygen, and nitric oxide. The point of my research is to establish the fact that when irradiated with infrared light, the dye will show enhanced photodynamic activity in the presence of DNA as well as in ovarian cancer cells. The methodology for this research included the determination of the specific wavelength or the range of wavelength under which ESS24 will display optimal photonuclease activity. The principal method used were UV-Vis absorption spectroscopy, DNA photocleavage, and photocytotoxicity experiments. UV-Vis spectroscopy experiment allowed us to determine at which wavelength the dye is most stable. DNA photocleavage experiment allowed us to determine if the dye can cleave the DNA or can it cut the DNA. Photo-cytotoxicity experiment, they allowed us to determine if upon irradiation, the dye will produce ROS in cancerous tissues, therefore producing toxicity to the cancerous tissues and killing it. When you take the root, photo means light, cyto means cells, toxicity means toxic or killing. So upon irradiation, are the cancerous cells or tissue being killed? The expected results were to find enhanced activity of the compound upon its irradiation, the mechanism and the type of ROS being generated upon photocleavage by the dye, as well as the precise DNA binding mode of the dye. We want to find the enhanced activity of the compound because we want to see at which wavelength is the compound most stable. We want to know the type of ROS being generated because we want to know, is it hydroxyl radical, superoxide, hydrogen peroxide, singlet oxygen, or nitric oxide being generated? Because of the previous studies and the pattern that my dye exhibit, we think that it is most likely hydroxyl radicals. We want to know the precise DNA binding mode of the dye, meaning we are trying to figure out how does the dye bind with the DNA. Does it bind to the minor or the major groove? Where on the DNA does it bind? As far as our results, the UV-Vis spectroscopy experiment showed that the ESS24 is stable in sodium phosphate buffer in the absence or presence of DNA, as you can see in figure 3 and 4. For these results, we can conclude that ESS24 was able to display minimal absorption at long wavelength value. However, when the dye is by itself and mixed with the DMSO solvent, it displays a much stronger absorption, as you can see in figure 2. For the DNA photocleavage experiment, you can observe in lane 2 cleavage, which is a lane that contains a sample that was made of the dye, the sodium phosphate buffer, DNA, GI water, and it was irradiated under the light at 830 nanometer at 10 degrees Celsius. However, when the sample was kept in the dark, no cleavage of DNA was observed, as you can see in lane 3 and lane 4. Lane 1 was just the control. It did not show any cleavage as well. These results demonstrate that the ESS24 is capable of cleaving double-stranded DNA under the given condition, which shows potential as a photosensitizing agent in PDT. In the photocytotoxicity study, the conclusion reached were that the ES2 ovarian cancer cells can take up the ESS24, as you can see in figure 6, and that the percentage of survival of the cell was greatly reduced when they were irradiated at 808 nanometer, as you can see in figure 8, compared to when they were in the dark in figure 7. Last but not least, there was a significant amount of ROS production when the dye was irradiated under long wavelength light, as you can see in figure 9. So in conclusion, our findings demonstrate that the ESS24 dye might be a potential candidate as a photosensitizing agent for PAT, as it would most likely produce hydroxyl radicals. I would like to give a special acknowledgement to my faculty sponsor, Dr. Grant, our faculty collaborator, Dr. Henry, and Dr. Torachula, as well as my mentor, Effie. I would like to thank the Honors College for allowing me to pursue research at Georgia State University, as well as the chemistry department. Thank you for coming and for listening.